I'd like to thank you for being allowed to speak today and talk about some of my work into the... Oh, okay. Thank you for speaking. Thank you for letting me speak. I'm going to talk about some of my work into the evolution of monoamine cell types. So, what are monoamines? They're a group of molecules. They include things, even if you're not familiar with them, that you'll be aware of, like serotonin, dopamine. They're present in all sorts of different organisms, but they are used in animals as neuromodulators, and they influence a whole range of neurological phenomena. We can see here we've got a series of biosynthetic pathways, and all the different monoamines are derived from amino acids through a series of different enzyme reactions. And a lot of the enzymes are either the same or closely related between these different pathways that perform these reactions. And the same is also true in their functional pathway. So we can see in the image of a synapse that a lot of the same genes are either or related genes are used in transporting and detecting these molecules as well. So we were interested in the evolution of this pathway, the genes, and then also how these genes are controlled and regulated and where these genes are expressed. But when we first started, a lot of the phylogenetics of the genes was still uncertain, so we carried out a phylogenetic analysis, which you can look at in the paper we published last year. And the broad summary was that even though monoamines are found in lots of different organisms, the canonical pathway, which I just showed you, has only been derived from animals, or from vertebrates specifically, and this pathway appears to be largely unique to bilateria, and all the specific orthologs originated through a series of gene duplications in the last common ancestor of bilaterians. And this is the group that we know uses monoamines for neuromodulation, with early animals so far not known to have this feature, and the only members of this pathway that they do have are enzymes that are used in lots of other things as well. So as I mentioned, we're also interested in how these genes are regulated, because in order for the actual pathway to function, all the genes need to be expressed in the same place and have this coordinated. So we're interested in what specifies these cell types and what turns on the different genes in these specific locations. And it's particularly interesting with monoamines because despite their large-ranging effects and influencing lots of different things, they're expressed in very, very small populations of neurons, which you can see in these diagrams above. And some of the work has already been done to identify which genes turn on the different monoamines in different populations, particularly in vertebrates and in C. elegans, and there's other potential ones that have been identified in other organisms as well. And in many cases, a lot of the same transcription factors and regulatory genes are coming up in different populations of cells in different organisms, and we're interested in whether this is significant, whether this betrays any deeper relationship between the control of regulation of monoamines or of the cell type identities themselves. So what we wanted to try and investigate was to see whether the monoamines are homologous and see whether there are any shared regulatory control, whether this is related to suggesting that the different monoamine cell types are perhaps derived from an ancestral cell type in earlier animals. And to do this, we're going to focus on single-cell transcriptomic data. For those who aren't familiar, single-cell is similar to normal transcriptomic data in that you're measuring expression through mRNA, but you're barcoding them, so you're able to extract out the expression profiles of individual cells and compare lots of different cells rather than a whole sample. But we have an extra issue in that we're trying to compare this expression across species, and the variables that we look at for each species will be the individual genes in those different species. And of course, different species have unique gene sets. And as you can see in the little sort of diagram of the gene relationships within species relationships, genes don't nicely follow how species are related. There's lots of different events, duplications, losses, that will make it hard to distinguish what gene in one species is comparable to another in a different species. Now, usually standard practice is that people will try and eliminate a lot of this and focus on just one-to-one gene comparisons, so either single one-to-one gene orthologs that don't go through any duplications or anything like that, or we'll try and select multiple homologs, filter them by similarity or something like that. But as in the little pictogram, this will reduce the overall amount of information that you're looking at for each gene family, and you will discard lots of data, not necessarily on whether it's biologically relevant, just on the fact that it's hard to compare. And this is only going to get worse the more species you compare, and the more distantly related they are, because there'll be more time for differences in gene set to accrue. So we used hierarchical orthogroups, which is where we add up all the gene expression for all the different genes that are within a gene family, not just a gene family in general, but particularly, as we can see in the little tree diagram, genes that are derived from a single ancestor at a particular point, a consistent point in species evolution. So it can be vertebrata or all animals or something like that, so that we have a consistent definition of the gene family and an evolutionary equivalent variables that we're comparing across the different species. We weren't sure whether this was going to ruin the signal in some of our data, but this is just an example data set of a sea urchin larvae. For those who aren't familiar with UMAPs, they're dimension reduction plots similar to a PCA, and the different dots are the expression patterns of different cells, and they're coloured by their cell type annotation. And you can see that in the original data and in the two different methods of grouping by gene family that we've attempted, we're able to recover the same cell type annotations. So it does retain the biological signal. So overall, for our project, we took a whole bunch of published single-cell RNA data sets. We took more than listed here, but we'll just focus on these for now. Took the equivalent proteomes, or the genes, annotated them to their gene families, recoded the single-cell expression data, and then we compared these and looked at the expression of the monoamine genes and then any potentially regulatory genes that are associated with them. So this is our integration. We started with a simple comparison of just several insects, because this is a smaller group rather than comparing all across animals, and these were all brain samples, so they're much more directly comparable sampling. What we're able to see is that, again, in this UMAP, the cells are colour-coded by their cell type annotation, and broadly, they cluster together by their cell type rather than by the species that they derive from, which you can see in the small insert in AII, that's coloured by species. So this is good. We're getting the cell type signal as opposed to any sort of species effect signal. And at the bottom right view, you can see that we've highlighted all the monoaminergic cell types were clustered in a specific cluster, cluster 20. So when we look at the expression pattern within this cluster, we see that all the different monoamine ortho groups are upregulated in this specific cluster in each of the individual species that we looked at. We were then able to do differential expression testing to see which regulatory genes are also upregulated and expressed in this cluster. In particular, we found one, the ortholog of prop1, which is significantly upregulated in all the different species in this cluster. And we then also able to subcluster and look at in more detail at different parts of it, so either just the serotonergic or just the dopaminergic cell types within this broad cluster, and we found even more different transcription factors which were conserved across multiple different species. So what this suggests is that there is some, at least within insects, conserved transcription factor regulation between the different monoamine cell types, and that all the different monoamine cell types within insect brains appear to be quite similar to each other, certainly compared to all the other cells in their brains. So we tried a more diverse data set using vertebrates. So this was older, more distantly related species, and we also included some other different types of data beyond the brain, particularly the gut, because several of these data had as important gut cell types in some of the mammals. And again, we saw that the cell types clustered by, the cells clustered by their cell type as opposed to by what species they were from, and in this case, the multiple different clusters of monoaminergic cell types. So in the bottom, there's several, the 22 and 62, which had most of the cell types in there, including, interestingly, not just the neurons, but also the gut enterochromaffin cells. There's also a distinct group which clustered with photoreceptors and also had a couple of extra enzymes, suggesting these were melatonin-producing cells. There's also a large group which had lots of different neurons and then a small proportion of dopaminergic cells in it, and these appear to be the olfactory dopaminergic neurons. And again, we looked at the expression patterns of different genes, and these match what we thought, and we tested and found lots of different transcription factors were co-expressed in all the different species, including those ones highlighted in bold, like DLX5 and FOX2B, which have previously been identified and are known to regulate monoamine gene expression in vertebrates. Interestingly, we found that in lots of different monoaminergic cell types and in those photoreceptor and melatonin cells, there's lots of overlap in the different transcription factors which were shared, except for that big, those dopaminergic olfactory receptors. They seem to use lots of a completely different set of transcription factors than all the other types. So to keep going, we want to look further, look across all bilateria. In this case, we're just looking at the different model organisms that we had. So this is from 4 different phyla. And we see that there is a beginning to sort of break down of the clustering by cell type, because these are very distinct or very different organisms and very different sampling for different data sets. But different cell types do still cluster together, like the muscles and then those photoreceptor melatonin cells, and also most of the rest of the monoaminergic cell types all ended up in a single cluster of 20, which again had upregulated monoamine genes from all the different species we were looking at. And we found at least 2 transcription factor groups which were, if not always significantly, upregulated in all the different species we looked at. So going on from this, we've been starting to do some validation of this. One of my colleagues has been starting to use HCR to map the mRNA expression of different transcription factors and the enzymes to see if they actually are co-expressed. In this case, we didn't mainly in Drosophila, we started in the embryos. We'll be looking at other ones as well, and hopefully moving on to some knockdown experiments. But currently, the picture that we found overall is that there seems to be a lot of similarity, particularly within groups, with lots of transcription factors shared, and within the vertebrates, there's lots of transcription factors shared between the different ones. But there appears to be some uncertainty between the exact direct relationships with nominally distinct cell types, like the gut cells or these photoreceptor cells, attaining some or co-opting some of the monoamine regulation pathway. But overall, across all bilateria, there's lots of different transcription factors which keep popping up in different phyla, in different monoamines, but it's not exactly clear how they relate to each other. They're just used in lots, the same set are used in lots of different ways across the whole of bilateria. So I'd like to thank the organisers again for letting me speak. Thank you all for listening. Thank the rest of my lab group who's helped me, and point to speak to my colleague Julienne, who had a talk earlier today. So I think that's the end. If there's any questions.
Hey, hi everyone. I bet you've all seen pictures like this one, right? They're beautiful. They're beautiful, aren't they? I mean, I do find them beautiful. I mean, they really show up how our own species, humans, distribute themselves over the planet and make up these cities. But they're also a little bit scary, right? Because like we do really get the visual of how much impact we have on our environment, especially when we build our big cities. And the World Health Organization now says that more than half of humans, even much more than that, now are going to live in urban environments and are affected by the living environment, but also everything that comes with it, right? And I'm a human geneticist, and human geneticists, we've been, in the last couple of years, putting together huge data sets of humans in different countries. And we have one of them in Canada that's called the Canadian Partnership for Tomorrow's Project, CanPath. It comprises about 300,000 different Canadians distributed across our territory. And we gathered information on these participants, going from clinical information, functional genomics, whole genome genotyping, and environmental data that's linked to them. You might have heard of other ones, such as the UK Biobank, which is one of the biggest ones. And those are beautiful resources that we can use to address eco-evolutionary questions. Also, these biobanks usually comprises of blood samples, but blood is a fantastic tissue because it goes all around our body. And usually humans, unlike animals that most of you studied, are not really willing to give out part of their tissues when they're still alive. So blood is good because people are usually happy to give it. And blood is made up of a range of different cell types that differentiate according to a very hierarchical process called hematopoiesis. And this is the type of cell types that we collect when we take a blood draw. And today I'll speak a little bit about a project, a long-going project about how the environment influences gene expression in our blood. And for that we use the population living around here in the Quebec province. And we sample from 3 different cities. So Montreal, which you're all familiar with at this moment, Quebec City, which is a smaller city a bit up north, and then the Saguenay region, which is a region, it is a smaller city. However, it is home to large industries, especially in aluminum industries. So the three cities have contrasting environments, and this is one interesting part to study. The second interesting thing about this population is that the large majority of the European descent population here, we refer to the population as French Canadians, that are originating from a colonization from France about 400 years ago, where the French settlers came first to Quebec City in a small number to populate the area, and then moved to Montreal, and then up north to the Saguenay-Lac-Saint-Jean region. And as population geneticists, we call this process sequential bottleneck with population expansion. So you can imagine people were traveling on these kind of buggies around 400 years ago. It wasn't easy to move across places, and this is what happened following isolation. You have population expansion, and this affected the genomics of genome of French Canadian with what we could expect, with a decrease in effective population size, higher genetic drift, and a higher frequency of certain deleterious alleles. So for our study, we collected, in fact, for this particular part of the study, we had 1,000 participants for which we had whole genome genotyping, bulk RNA sequencing from the blood, health history measures, and linkage to built environment and air composition measured from every of these participants. So the first thing we confirmed from other studies is that if you do PCA on the genotyping data, you could tell apart French Canadian from French people living in France, where they have slightly different allele frequency, and they show up as a different grouping on a PCA plot. But you can even go further than that. You can pick up those founder effect, where you can tell apart people that have a regional ancestry from Montreal, Quebec, or the Saguenay-Lac-Saint-Jean region. Interestingly, we pick up a similar trend in gene expression as well, where if you perform a PCA on this whole transcriptome in the blood, and you plot the PC1 value on a map, you do pick up differences between Montreal, Quebec, and the Saguenay region. So now the question becomes, is this gene expression decline mostly driven by different genetic ancestry, or is it driven by differences in the environment? So to address this question, we actually use our population like an experimental lab, where we first identified people that we call French Canadian locals that have a regional ancestry that's matching the city where they live at the moment. We have a second group that we call internal migrants, which are people, for example, that have a regional ancestry in the Saguenay-Lac-Saint-Jean region, but they're found today living like their houses in Montreal. And we have a third group where we have Europeans, so people who clearly have a genetic ancestry coming from Europe, but they live in each of the three different cities. So the first comparison we did is compare people that have the same regional ancestry, but they live in different environments. And I'm going to just show you a couple of volcano plots for those who are familiar with. For the ones who are not, every square is the comparison, every dot is a gene, and whenever you see a red dot, it means it's differentially expressed between the two conditions. So for example, here we're comparing Montreal locals, so Montrealers with a regional ancestry from Montreal versus people with a regional ancestry from Montreal but living in Quebec City. So we're comparing environment, people with the same ancestry. So in all these comparisons, we see a lot of differentially expressed genes. However, when we look at people that have different regional ancestry, but they live in the same environment, what we found is very little differentiation on the gene expression level, meaning that they have very similar expression pattern in the blood. And the last comparisons were European living across the Quebec territory, where, again, they are not differentiated a lot from French Canadian living in the same cities. So this indicates that the environment seems to be driving the expression more so than the regional ancestry. So when you plot that on a graph, on a heat map, just looking at the average level of expression from each of these groups, you get grouping according to the environment where people live rather than their genetic ancestry. And what's interesting is that we found an enrichment in these genes that are differentially expressed across region in gas transport and exchanges and inflammatory pathways. So this led us to ask the question, what could be driving this pattern? So with an amazing postdoc in the lab, Favé Lamas, we perform a co-inertia analysis. I won't go into the details of it, but we basically found that those genes, when we look at which environmental factor it could be driving the expression pattern, it was mostly associated with air composition measures. And also, if you look at clinical features associated with this gene expression, it was associated with arterial stiffness and cardiovascular diseases and inflammation. This is actually a pattern that's been found across different systems in the world where people live in urban environment versus rural environments. So the next thing that we did is that we asked the question whether individual genetic variation can affect the response to these exposures to the pollution. So we perform expression quantitative trait locale mapping, which is a statistical analysis when we look whether a genotype of a particular SNP is associated with the expression of a nearby gene. So for example, here you have a graph where you would have the genotype, homozygote reference, heterozygote, and homozygote derived. It would associate with increasing level of gene expression with increasing number of alternate alleles. And what you can do as well is to you can model whether you find differences in this relationship between the genotype and the expression across different conditions. So for example, the red boxes would be the relationship you find in an exposed group of people of a certain pollutant, and the orange box plot would be the relationship you find between the genotype and the gene expression level of not exposed people. So we performed that analysis across our pollution exposure. So we have 4 different pollutants. And we did find a number of genes for which the expression would be associated with a SNP, but also modulated by the level of exposure every group would have. So I won't go into the details of that, but we also recently went a little further and looking into a related cohort of CanPath, which is the Ontario Health Study, where we have 400 participants with single-cell RNA sequencing. And we asked the question whether we could find those same eQTL into our single-cell data and in which cell type they would be most present. So this is more like a future step to look at, but what we found is that this particular eQTLs that are associated with the response to pollution of individual, they seem to be enriched in adaptive cell types. So adaptive cell types are things like B cells and T cells. So these are the cells that learn to respond to immune stimuli and retain this memory. They have a lot of different functions, but these are known to be particularly important in diseases like asthma. The last thing that we looked at are cytokine levels. So cytokines are inflammatory proteins that they're like messengers. So they help immune cells to talk to each other in close proximity or even further away in your body. So they're very complicated and they secrete a lot of things. But what we did is that we have panels of cytokines for about 8,000 participants across the Canadian study, and we do have the information of where those people live. And we started looking at, so this is just an example, this is Montreal, this is where we are at the moment, and every little square that's colored is a neighborhood for where we have at least 50 participants where their cytokine level measured. So this really opens up the possibilities to look at very precise level of environmental exposure that could be modulating this protein expression. And what the first thing we try is to see whether we can reproduce our finding that we found with the gene expression. And indeed, even with a small number of proteins, we do find some of the, for example, in the NO2, which is forget the name, NO2 exposure, we do find some proteins that are dysregulated between people that are highly exposed versus people that are not exposed. And these were also, we find the same matching gene in our gene expression data set that is also dysregulated by NO2 exposure. And we perform the same for PM2.5, which are small particles usually emitted from transportation and industries. So hopefully today I show you that human is still an interesting species to look at. and that the different and burn environment can strongly affect our gene expression of our immune circulating cells, that the individual genetic variants may influence the response to exposure in a cell type specific manner, and that downstream markers such as protein levels also show dysregulation with pollution exposure. And I take this opportunity as well to say that I'm going to start my own research group in computational genomics next week. So if you're interested, you can come and chat with me for any graduate student possibilities. I will also be looking for a bioinformatician. And we will look at anything related to eco-evolutionary medicine in large population cohort, including spatial transcriptomics, building risk score, a bit more medical, but building disease risk score, but also looking at human archaic entrogression and whether those influences disease risk scores. And possibly also to come back to this beautiful picture at the beginning, using satellite imagery to better characterize the environment. So with that, I would like to take the time just to thank my lab mate of the last 10 years in the Wadala lab at the Ontario Institute for Cancer Research, and I thank you for your attention.
Hello. So I'm Jason Keagy. I'm at Penn State University, and I will tell you about a landscape transcriptomics approach where we're trying to evaluate and predict organismal response to extreme heat events. I'll lift that vague as to organism. That'll be the next slide. But I also want to call out Justin Warniak and Tyler Wagner as my collaborators on this project. Okay, 2 slides, you'll see what I'm talking about. The big picture context of this is heat waves, and these are just some pictures from this summer and last summer from various news sources showing this as like a global problem that we've probably have experienced. Like I experienced this one, I'm not sure if a couple weeks ago down here. And so heat waves are one consequence of climate change that is a lot of times the global temperature increase, and some of those knockdown effects are really focused on, but these are actually maybe in some ways even more consequential because you have a dramatic increase in temperature over a short period of time that is basically defining the heat wave as being above average. And these are increasing in intensity, they're also increasing in their length and how frequent they are. And so we are having trouble dealing with this. Like this is a big health concern for humans. It's also, like economically, it's a problem. Organisms are as well. So here's what I study right now. Is the brook trout. It's a very beautiful fish, I think. I'm not actually a fish person, honestly, even though I've been working on them for a while, but I think that's beautiful. There is salmon that lives in the eastern North America. Most of them are, I would say, there's a lot of really cool life history and different ecomorphs and stuff. We're focused on stream-dwelling ones. So these are living in these really cold headwater streams. Stream temperature is a really critical predictor of whether they're going to be in a stream. And they get these physiological consequences at 20 to 23 degrees Celsius, which you'll see that the streams that I'm going to talk about, we get above that. And so we're very interested in knowing more about this thermal stress response. There is work on this because people, for example, eat salmon or they like to go fishing for different salmonids, and so there's a lot of interest in this problem. So there's lab work on this, and people have been working on focus on certain physiological measures or very specific genes, but it's generally in the lab, and so we really wanted to ask the fish, like themselves, like in the wild, where they can move around and do stuff, and I mean, it's kind of a mess from an experimental point of view, but that's really what we want to ask them directly, like, how are you responding to this thermal stress? What's going on in your life? And so the tool we're using is transcriptomes. I guess the theme of this session, really. And so we'll need to say too much maybe about this, but we're going to go and actually look at the mRNA in a given tissue at a given point in time. That given point in time is actually really important. And we can look at things like expression level, other genes that are differentially expressed. And here I'm comparing populations, for example, or you could like try to take that information about the whole transcriptome and how things are correlated and try to infer gene networks to get like a higher level view of what's going on. And so this gives us lots of information. It will provide insight potentially into molecular pathways associated with the stressor. I also I'm working with trout. I do some of this has a nice applied aspect to it, and that is that gene expression is a phenotype. And so this could potentially be leveraged as a stress biomarker. And this is a shameless plug for a paper in Molecular Ecology Resources where we really wanted to, there's lots of people, landscape genomics has existed for decades, and there's lots of great review papers on that. And often nowadays, there'll be like a tiny little paragraph on transcriptomics in those review papers, because everyone is at least aware that transcriptomics exists. But we thought a whole paper was needed for this because there's lots of potential pitfalls but also promise from using transcriptomes that you gather from a landscape. And so, shameless plug. But we're going to look at 2 different studies. And the first one was focused on all these streams are in the same watershed. This was interesting because it's something that the Fish and Wildlife Service has been monitoring for 20 years. We actually know a lot about the kind of history, the thermal history of these streams. And here's some pictures of the beautiful locations. But we went out and we only sampled one time. That's going to be really important to the study design. We just went out in the morning. The fish that should not have been, we were catching them in water that should not have stressed them out at that moment. And we just went out there and grabbed some fish. We working on gill biopsies because unlike humans, or maybe it's just me, they're not happy when I get blood from them. I'm just not very good at it. The gill, they give up fairly readily, and they'll grow that back. And so we take a little snip of that. It's not lethal, which is great for the fish, so I'm sure they enjoy that part of it. But it's also a tissue that's really important for thermal stress. It's got some really interesting biology and physiology. It's in contact with the water. This is showing my hand there, so you can see it's not that much tissue, and we can get RNA and DNA from that, and then we can, for now, mostly what we did with that is just sex the fish, but from the DNA. The RNA we used, the particular sequencing strategy was 3' tag seq, and we did that for 6 individuals per stream. And then we can ask questions are the populations differentiated? Now, these populations do have they're all connected in the same watershed, but they can get quite isolated. They have different temperature kind of history. There could be some cool local adaptation. We can't actually get at that with this study design, but we might still see some signal of population differentiation. We just won't know why. Those streams that usually generally are warmer were not necessarily the warmest streams when we sampled them. So we did break that correlation a little bit, at least. We could also ask if there's sex differences, and then the big question was really, is there going to be an association with temperature and gene expression? So very briefly, this is a principal component analysis showing the gene expression, the 2 major axes of expression. You don't see clusters of color, so there wasn't a lot of population differentiation in this study. When we were looking at that single time point, we didn't see gene expression differences by the sex, but we did see. And honestly, I was a little surprised by this. I know you're supposed to go out and do studies that you know you're going to get some great results, but I thought I'd see some genes, but we saw quite a good number of them. And then these are candidate genes, which was validating for me that some of the genes that people were finding in the lab were also finding in nature. And then, what I think to understand the context of this, 13 degrees to 17 to 18 degrees here, these fish they shouldn't have been stressed at that moment, but they probably got stressed the day before, and they may have also, physiologically been anticipating getting stressed later that day. So that's what we think mechanistically is happening here to get this signal. But we've got 2 heat shock proteins. This is a general kind of stress response protein, and then this one's actually involved in cold stress. So we get down regulation there as it gets warmer. So we thought that was interesting, but it's limited because we're just looking at one time point. So then we went and did this study here where we had 4 streams that were much closer to our home in State College, and we went out and we sampled 4 times during 2 successive heat waves. And I guess just to paint a picture of how insane this is, determine a heat wave, we were just looking at weather reports, which aren't great, especially in our area, and trying to predict when there would be not only 1 heat wave, but 2 heat waves, and then go out there and sample. It would have been okay if the heat waves were a little bit further apart. These happened to be right after each other. But then we went out and just kind of hoped that things would work, I guess. But these are temperature, it did work, because these are temperature profiles in the water. And we did go out and do some pre-scouting. So we were trying to find 2 streams that were kind of warmer and more variable, and 2 streams that are a little cooler and less variable. And there's this nice double hump here, and we were trying to get the ascending limb and near the peak, and the descending limb, and then like a week after the peak, and then repeat that. And so that's what we got. And so we have 8, from 4 streams, we have 8 time points. They're not the exact same fish, they're just the same population. The first hint that something really cool was going on is that this is just taking one of those candidate genes and plotting the gene expression, and you can see that they have 2 humps. And that's the temperature profile there, showing 16 Cs where they're really happy, 20 Cs where they start getting really unhappy. And we see these 2 humps, and it's kind of stratified by the population, by their temperature profiles. And so then we took a much deeper look. This is where Justin Warniak came in. He did some really creative analyses. I'm going to show you one because I'm going to run out of time, I'm sure. So I'll just show you one. He used this package called Impulse DE2, which it's really designed to look at time course studies, because those can be a little challenging because you have autocorrelation between your time points. So this is modeling that, and then it's looking for genes that, and what I'm showing here in the figure, I probably should have animated this because that's not what Impulse DE2 is doing. That's something different. But Impulse DE2 is just looking for genes that are changing over a time course. And he identified 43 from that analysis that were associated with time, and most of those were actually associated with temperature as well, because we have 2 things going on, right? We have time, like the seasons dragging on, and then we also have that temperature is increasing and then decreasing. And so here, now what he's done is taken these genes, well, I just took the 3 that I showed you earlier. He's got temperature, and then the expression, and you can see here that we have this these genes, just like in the first other study, they're getting upregulated with temperature. And we also know, because they were identified with that method, they're changing over time. And then there's one other one I threw on here because this is really popular with the cell-mounted people, this heat shock protein 70, which we didn't see in our previous study, but that's only usually expressed in the lab after they're like near death. So yeah, there's that. And then I'll just summarize really quickly. In this first study, we didn't see population differentiation, but we did in the second study when we started looking at this time course. Didn't see sex differences in either one of them, and then we found like some pretty cool like associations with gene expression and water temperature. And basically what we're trying to do is like take multiple approaches. So coming up next in a week or 2, we're going to be using these experimental stream systems, and that's in this big warehouse in West Virginia, where we'll be able to actually ramp up temperature and try to triangulate on like some markers of stress. And hopefully, if we get the money for it someday, we'll be able to do more of a continental-wide kind of approach and start looking at actual questions related to local adaptation. And then this is like the kind of applied implication. I think I'm actually feeling really positive about, and this is not like my idea. I mean, there's other people, this is a really great paper on this topic when fishes, but we could use these we've got lots of state and federal partners who would love to be able to use this to assess response to changing environments in real time, and also to identify populations that are stressed or not stressed, especially because people like want to move these fish around now because of climate change, and so maybe we can pick the right ones to move for assisted migration. And so I will leave that acknowledgement slide up here. These Sarah, Julie, and Natalie were instrumental in that crazy second study field work. They're the ones who really made that magic happen, and we have a partner, John Svika, U.S. Fish and Wildlife, and then we have various sources of funding. Thank you. Yeah.
Do you think you'd see the same results with no differences between sexes and populations, like depending on what time of the year you're sampling them? Or do you think it would be the same year round?
Yeah, that's a great question. We are going to be addressing that question in a different species, brown trout, actually, this year. So we just started our summer sampling. But we are interested in seeing other stressors too, like reproductive stress, and trying to sample at a time when they shouldn't be stressed, and kind of comparing and see if there's different sex differences and things like that. Yeah, and I think I probably used up all my time. So thank you.
Good afternoon, everyone. It's now afternoon. So I am Anna Savage, and I have been at the University of Central Florida for almost a decade now, but the data I'm going to present to you today are actually samples that I collected during my Smithsonian postdoc in the early 2010s. And the analyses I'm going to present to you are brand new, really cool work done by one of my graduating PhD students, Nick Christodoulides, who has led the bioinformatics on this project and turned this old project into a dissertation chapter. So I'm very happy to finally be able to present these data to you. So as an evolutionary biologist, I love that we get to ask the question why. You know, one of the things that drives my science is really wanting to know why things are the way they are, and we get to do that. And as an amphibian biologist, I love that I get to ask a lot of questions about why different frog populations do or do not adapt to certain stressors, and why amphibians are dealing with such massive disease problems. And so a lot of my work has been driven by asking those questions and hinting at some answers, maybe getting some answers, but actually where I think it's led me currently, and maybe some others in our field, is that the questions of why have led to even more questions of how. And it's made us realize that a lot of the mechanistic underpinnings of how frog immune systems work, for example, are really poorly understood. And so I'm here at Evolution giving to you a pretty mechanistic talk that hones in on the how in order to help get better answers to the why. So as I hinted at, the study system that I focus on is amphibian chytridiomycosis. So you're seeing some of the images that I experienced chytridiomycosis as in the southwestern United States. This is a disease that's specific to amphibians and has caused declines and extinctions in hundreds to thousands of different species. And the culprit behind chytridiomycosis is largely Batrachochytrium dendrobotatus, a chytrid fungal pathogen that I'll just refer to as BD for the rest of my talk. So one of those big why questions that my research program has asked about chytridiomycosis is, why is it that different host species are very different in their susceptibility? So you can look at a scale from completely susceptible all the way to completely tolerant, and I've thrown up just different closely related members of the genus Rana in North America, and they fall everywhere along that spectrum. So there's huge variation at a shallow evolutionary scale. And one of the species that I've really honed in on is Rana yavapaensis, which was my focus of my dissertation, and I've continued to study it because it falls on the kind of more susceptible side of things, but shows a lot of intraspecific variation in susceptibility. So this Rana yavapaensis, the lowland leopard frog, is endemic to the southwestern United States as well as Mexico, but I only study the United States populations shown on the map. And this is a species that we know has been dealing with BD die-offs for 40 years, and the pathogen probably considerably more than that. And so what I'm showing you on the map are symbols with colors representing distinct genetic lineages that do not have any contemporary gene flow. And the different symbols represent the different susceptibility categories that we've established, where some populations are entirely tolerant to the pathogen at present, others remain susceptible, and then we also have populations that still show individual variation in susceptibility, so they are variable. And then the last thing I need to mention here is that there is one population shown in green that's sort of special because these frogs live in a thermal spring year-round, and BD does not like it hot. And so we call these frogs sort of environmentally sheltered from the pathogen, so it's there, but it doesn't really do much to them. And so we sort of consider them as a control population of what these frogs look like in a pre-BD world. So as I mentioned, I've spent a lot of my career looking at Rana yavapaensis and BD susceptibility, and so we do know that there are immunogenetic as well as transcriptomic predictors of BD susceptibility, but within and between these populations. And in particular, what we're finding is that genes of the adaptive immune system show both expression and allelic associations with disease susceptibility, and this is largely based on controlled lab experimental studies where we can kind of tease out that genetic component. And the pattern that we predominantly find in these lab studies is that if you are expressing more of these adaptive immune genes and sustaining that expression for longer, we actually see a lower survival probability. So we're seeing a pattern where potent immune responses with lots of inflammation seem to be negative for survival. So that leads me to my talk today about wild transcriptomes, and our argument here is that even though doing transcriptomics on wild populations can be messy, it might be key to understanding really what's going on with natural adaptation to BD. So in terms of this disease, there have not been any prior gene expression studies sampling wild individuals in situ with the disease and trying to tease apart what is the functional basis of BD survival. It may or may not match what we're seeing in controlled lab experiments, given that there's so much environmental variation in this system. So our challenge was to try to isolate what we could attribute to disease processes versus gene expression that we might find based on climate, sex, behavior, microhabitat, all of these other things that are going on. So to bring it back to my map of my Arizona populations, our research approach was to sample these frogs in situ in both summer and winter, because this disease is very seasonal. You only see chytridiomycosis in the winter. And so we had a sample size of 56 frogs across these populations with different susceptibility categories. And we sampled skin and spleen tissue because BD only infects skin, so that's the site of infection, and spleen is a major immune tissue of amphibians. So we also took swabs using a standard protocol for quantifying BD infection intensity, so how heavy is the pathogen burden on each individual. And then we included sampling day, those susceptibility categories across populations, the BD intensity, and interaction terms to try to tease apart BD processes from other natural gene expression variation. So we used a fairly standard Trinity-based approach to generate de novo transcriptomes because we do not have a reference genome for this species. And then we had sort of two major analyses. The first was differential gene expression using DE-seq2. And our design was to include the susceptibility category of the population, the sampling date, the log transform BD intensity, and then the interaction term that ended up being very relevant was the susceptibility interacting with the BD intensity. We also did gene co-expression network analysis using WGCNA, and then we took those modules and looked to see whether or not we saw significant expression patterns that related again to our disease metrics as well as our sampling date. So to get into the differential expressed gene results, what I'm showing you here is a Venn diagram of the overlap and non-overlap between the significant DEGs across those 4 variables that we ended up including. So the first thing I want to highlight is in green sampling date. We have over 1,200 DEGs that are uniquely assigned to that variation in sampling date, which was not surprising at all. What was a little bit more surprising was that our largest group of unique DEGs was actually that interaction term between BD intensity and the population susceptibility category. So that ended up being a much bigger component of the DEGs than we maybe thought we would find. And then when it came to just BD intensity on its own and susceptibility category on its own, there were very few to no unique DEGs, but there we weren't that surprised given that these frogs evolve BD tolerant, so they actually tolerate infections, and so that's where we did expect to see interactions going on between the intensity values and the susceptibility category. So now I'm going to show you the much messier heat map version of all of our DEGs, and so here you've got yellow being increase in expression and purple being the decrease in expression, and the vertical columns are each of those individual frogs, and all of the stuff at the top and the bottom are all of our potential groupings based on susceptibility category, our BD intensity values, our sampling date, some other things we kind of threw in there. The big picture I want to highlight is that there are 3 clear clusters of frogs showing differential expression patterns. They are not clearly and cleanly explained by any one of our variables. But if you look at the biggest cluster of genes in the top right, the ones that are showing higher expression of those genes, these are individuals that are mostly from susceptible populations or variable populations, and they are the frogs that had the highest infection intensities. So overall, they hint at what susceptible frogs are doing. And if you look at the gene ontology terms associated with what those genes are, what we see are adaptive immune genes, T cell, MHC, and actually the biggest chunk of those genes was inflammatory response genes, which are again those mediators that are associated with larger magnitude of immune response. So this was interesting in that it did largely match the patterns we'd found from previous lab studies where that adaptive immune response and inflammation was associated with the negative survival outcome. Okay, so moving on to our WGCNA modules, what we found here is we had 30 modules in total, and most of them we couldn't tease apart the environmental factors from the disease factors. It was kind of a mix. But 7 of those modules, the ones that I'm showing you here, were the ones that were not at all related to sampling date and only were relating to BD metrics. And so the top 6 of those 7 modules all showed basically that same pattern that I showed in the overall DEGs, where the bright yellow cluster of genes were the ones that were in the more infected frogs from more susceptible populations, and they included a mix of immune genes as well as a lot of other genes that I could talk about forever, but I won't. What I do want to mention here is that this bottom cluster was the only module that actually showed a pattern of the more tolerant frogs that had lower infections, showing a higher gene expression level. And when we look on the plot here at what are those genes, ranked kind of from larger numbers down to lower numbers, and the colors are the significance levels, we see basically gene regulation, a lot of regulation of gene expression. So hinting at a more modulated type of response going on in those frogs. So the last thing I want to show you is going back to the straight differential expressed gene results. And what I'm showing you here is we pulled out only those genes that had dedicated immune function in terms of their gene ontology. So you're just seeing the gene expression on the y-axis, and then the x-axis is that BD intensity value going from uninfected to higher levels of infection. And we parsed it out across our control populations, susceptible, tolerant populations, and then those variable ones. And in the control and susceptible populations, what we see is that as you start out uninfected, there are lower levels of immune gene expression, and then as you go through low levels of infection to progressively higher levels of infection, you see the ramping up of the immune gene expression. So matching that idea that if you're sustaining those potent immune responses, that's associating with susceptibility. In the tolerant populations, we see a very different pattern, which is that you see when you go from uninfected to low levels of infection, that's where you see the biggest spike in immune gene expression, so sort of early expression. And then as you go to higher levels of BD infection, you see a decrease in the immune gene expression. So sort of, again, matching this pattern of modulating immune responses if you're going to survive. Variable populations kind of show both patterns, and we see both categories of individuals there, so that kind of also fits. So to try to kind of summarize, as disease gets worse, what we think we're seeing is that immune genes are decreasing expression in tolerant populations, but increasing it in the control and susceptible, and then we see both patterns showing up in variable populations. So to kind of summarize and hint at significance, we do think that this is the first study of wild BD gene expression work that does hint at patterns that are consistent with what we see in the lab. So despite all that environmental variation, in the field we see that as infections get worse, we see that the frogs that are able to survive actually ramp down their gene expression, whereas the frogs that are susceptible are having these massive inflammatory adaptive immune responses. And so I think that this type of work is messy, yes, but important for really trying to understand how BD is going to impact natural populations of frogs that are what we're really concerned about. And so with that, I would like to thank my funding sources, which is largely Smithsonian and a lot of field people and lab people. And then I wanted to call out my student Nick doing his power pose at his PhD defense a few weeks ago. So he successfully defended his dissertation, including this work, and moving on to a nice bioinformatician job. And before I fully wrap up, I did just want to put in a plug that I am recruiting. I'm looking for some graduate students who may be interested in working on evolutionary trade-offs related to immune function in frogs, as well as just direct metrics of immune function. And so get out the word if you know of anybody who is interested. Thank you very much. Yes.
Hi, great talk. I was curious, are you categorizing these tolerant versus susceptible populations as that of a multiple functions? And if so, do you think it's something that is like genetically coded or possibly genetically is related to the adaptive?
Excellent question. Oh, thank you. Excellent question. So when I kind of glossed over the susceptibility of these populations, that is based on field work as well as lab work that is talking about contemporary susceptibility, right? So do we know for sure when and how it evolved? No. Is it standing genetic variation? But we see genetic allelic associations with those susceptibility categories in the lab that match the field. So it is a little bit indirect, but we have a pretty good amount of data to support that it's the current susceptibility state, and that it does have a heritable component. Thank you.
I'm so excited I got to see these talks, although you might look at my talk title and be like, wait, that's not gene expression. We're actually still waiting on the data that I was going to present, which was about gene expression under nitrogen source in this cool species. Instead, I'm going to tell you a little story about methylation, which I actually think is sort of relevant to the last couple talks, so hopefully you'll find it interesting. I want to say there's lots of folks who contributed to this, and specifically there are 4 folks who are not only in the room, but also presenting. They're outlined in white here. And I'm going to highlight what they're going to talk about. UMass Boston's a great place to work and teach. I'm talking about Salicornia. Salicornia are weird little guys. They are highly tolerant of salt, halophytes. They live in places where other halophytes can't grow. And they don't have leaves. They're kind of like little sticks. They're sometimes called sea beans, sea asparagus, samphire, swamp fire. The French-Canadian name is rude. So I'm not going to say it, but if you're interested, I can tell you. And has anyone in here eaten them? You can eat them. Yeah, they're tasty. You maybe got them as like a side, as a vegetable if you live in the UK. You can harvest them actually here in Montreal. They grow in near here. But they like to do lots of things, including tolerating and accumulating in their tissues at really high concentration heavy metals: arsenic, cadmium, copper, nickel, lead, vanadium, zinc. They volatilize selenium. At least other members of the genus do. And most importantly for me, they grow right near my campus. And we work on this species, Salicornia depressa, the common glasswort. It's the most widespread annual species in North America. It's on every coast and some of the inland places. And one of its ecological roles is that it colonizes bare patches and salt marshes. There it reduces local salinity and then facilitates other plants moving in. Why do we care about it? Well, like many places in the world, Massachusetts has a long history of human industry and lots of people who live there. And there are lots of toxic sources of different kinds of things, but in our case, we're thinking about metals in the state where I work. And those metals, they often get into watersheds. They flow down watersheds. They end up right there at the sort of interface of fresh and salt water. They tend to fall out of solution, ending up in coastal and estuarine and salt marsh sediments. So they're there where these plants that are tolerant of them are growing. And so we wanted to know, what's that doing to the plants? How are the plants changing that, if they are? So one of the first things we did was we went out to some marshes where they grew, and we characterized the lead in the sediment underneath where they were growing. This was an honors thesis by Philip Stefanovich, and we found that there's variation. So there's some things that are like toxic. Don't eat the Salicornia from here, levels of lead, and then other places where there's not a lot of lead at biologically relevant levels, at least. And these 4 marshes in particular are the focus of what we're going to talk about today, but they come from Boston Harbor. Do not eat Salicornia from Boston Harbor. Down here, sort of in the armpit of the Cape, or like the elbow, I don't know, that part of the Cape, where there's a National Estuarine Research Reserve, or Quake Bay. And then 2 sites on Nantucket, one right in the Nantucket Harbor, which is more impacted than this 150-year-old preserved marsh. That's where the Nantucket Field Station is. If you want to know more about what lead does, phenotypically to this species, you can go check out a poster this evening, by Scarlett Tavares Guzman. Okay, one of the first things we did in studying this species, it hasn't really been looked at before, so we assembled the genome. If you want to see a really cool talk this afternoon, you could go and see like chromosome scale, tetraploid plant genome assembly, learn some hot tips by Cooper. But one other thing that Cooper did in sequencing the genome was to use one approach, Oxford nanopore long-read sequencing. That gets you really long reads of relatively low quality, but they're super long. But one of the other things you can get from those data, as the DNA are being pulled through the pores, those nanopores, what's being read is sort of the signal, the electric signal, as it gets pulled through. And actually that signal is different when you pull through, say, a cytosine that's not methylated and a cytosine that is methylated. So you can use that signal information to determine methylation state, not only of cytosines, but also adenosines, which is more than we have been able to do with, like, say, bisulfite sequencing, and also much cheaper and ecologically scaling potentially, which is quite cool. So Cooper not only did this for the nanopore, the genome individual, but also got funding to do it across wild marshes. So populations at those 4 different marshes, 12 plants at each place, we ended up with about 3x coverage, I think. I hope I'm right about that, at least in our final data set. Okay, so we have methylome and genotype data, genome-wide, for 12 plants from each of those different levels of impact locations. And these data are really new. I just analyzed them this week when I was desperate because I didn't have the RNA-seq data. But I think we have some cool stories to tell. The first thing is there's structure there. It seems to be population level mainly. The big thing is that the stuff north of the Cape, which is the Boston Harbor site, the highly impacted site, is very different from the other 3 sites, which are all below the Cape and closer together physically. That site that's most genetically distinct is also significantly more inbred, which is maybe because it's more contaminated, but also it's smaller and more distant, and we have relatively limited population level sampling, so very hand-wavy there. And if you want to learn more about population genetics and Salicornia depressa, I highly suggest tomorrow you go to Alice Palmer's talk, Sea Beans in the City, Revisited. But what we can do with these data is not only look at genotypes, right, and genetic structure, but we can compare that to methylation differences, methylation variation, methylation structure. And this is what we see. So one thing I think is sort of interesting is it's much less structure, which you might expect, because the methylome both potentially is genetically inherited, but also or genetically encoded, I should say, but also can change with the environment, just like gene expression. In plants and also in vertebrates, methylation tends to be relatively limited, and it tends to actually be correlated with increases in gene expression, the opposite pattern of vertebrates, right? So where there is methylation in plants, you tend to expect to see increase in gene expression, just saying that in case you don't know. One thing we do see, and this is really like exciting, suggestive, but it does not show anything, is that the most different populations in our first 2 PC or scale dimensions, this is an MDS plot, are sort of the least contaminated site pulling up this way and the most contaminated site pulling down that way. Maybe there's a pattern there. We're going to have to follow up and think about it more. So even though there isn't clear population structure here in the same way there is for the genetic data, we wanted to ask if they are related, if we can predict methylation state from genotype or from genetic relatedness. I wanted to point out really quickly that little weirdo there. This is one plant that's doing something different from everybody else. We don't know why. But, and we found that, indeed, genetic kinship measured from these 2.3 million SNPs is predictive of methylation similarity. This is a Mantel test of the two matrices of relatedness from the 48 samples. And that's kind of cool. What else we can say from it? I'm still thinking about it, and I would love for you to tell me what you think about it. One thing is that little weirdo is down there. And then, like, what's going on here? These are some highly related plants that don't sort of follow the general pattern of being more similar methylation levels. Maybe there's some sort of, who knows? I could tell some stories. I'm not going to. Okay, so short and sweet, I think we're going to get to lunch early. But one thing that I want you to take away from this is we have been doing transcriptomics for a long time. We've sort of democratized the platform so you can do it in the field. We saw 2 really great examples of this, of people doing transcriptomics in the field. And I think we can also do this now with methylation sequencing. Bisulfite and equivalent approaches are really expensive. They take a lot of sort of lab know-how, or you can pay for them. But we are able to do this for like 4 flow cells of nanopore, which was, I don't know, like $3,000 something, plus the like library prep stuff. But we could do 48 individuals for, let's say, $4,000. And that's great. And we have a relatively big genome. If you have a small genome, you could even put more on there. So I think the potential here is to start looking at other levels of variation, right? We have genotype, we have transcriptomes. What's sort of informing that relationship? Maybe there's epigenetic stuff we can look at. And then, of course, maybe we can someday democratize the proteome, maybe. But so insight, I mean, I don't know. I think we might have needed better sampling in this case because we can't disentangle the contamination from the genetic differentiation in our sampling. And we can do that because it's relatively cheap. If you want to talk to me about how we did this, I'm happy to chat. And also if you want to talk to me about like what I should do next, please let me know. There's one other person, Emily Mello is going to be giving her poster on Monday. That's her third time doing it, her honest thesis. It's also not really gene expression because we don't have the data yet, but there'll be some phenotypic patterns and some example analyses. Okay, and then finally one we're talking go to with the same sort of approach in a animal. Ooh. Is also a poster today. Thank you so much. And I'm pretty sure I have time for questions. Yeah, if you want to ask a question, you can come chat with me, or you can head out. Or go ahead, Isa.
I'm going to ask a question.
Yeah.
You mentioned that nanopore is a little bit error prone.
Yeah.
Is it more or less error prone for methylation?
That's so. I think it's the same amount. But I mean, I wouldn't.
Okay.
I don't know if that's. I don't know. Actually, is my answer.
Okay.
I wouldn't say it's more accurate.
Yeah.
The thing you get is just you get genome wide. There's some you can use like some deep learning models to sort of build models that predict, and we use the sort of the most basic algorithm that's built in already to the Dorado base caller. But I think the power is in the cost effectiveness, not necessarily in the perfect accuracy.
Yeah.
But at population level, that's fine. I think if you're worried, if you're looking at a specific mechanism, you might want to confirm with higher.
Depth.
More validated.
Oh.
Higher depth also,
Yeah,
yes.
yeah.
Through
Okay.
3x. But we could get information with 3x, which is cool.
3x, okay, thank you.
Yeah. Thank you. Thank you all.